Hi, I'm Viviana Gradinaru from Caltech. The title of my talk is Blood-Brain Barrier, Friend and Foe. Thank you for the opportunity to interview for the HHMI Investigator competition. My dream is to understand the basic biology underlying physiological defense structures, such as the blood-brain barrier, and in particular, the mechanism of transport across the blood-brain barrier, which could also help us move towards targeted, non-invasive study and repair of the brain. What focused me at first on this problem was my prior work with neurotechnologies. In neuroscience, it is an exciting time with all the genetic and optical tools, including the ones I worked on, such as optogenetics or tissue clearing and imaging. But this work also taught me that we do have a major problem in accessing circuits that underlie both natural and disease behaviors, because they do involve distributed, molecularly diverse circuits throughout the brain. And in order to understand and restore brain function, we need brain-wide access to deliver genetic tools and therapies. How can we gain such access, especially to deep brain regions? It turns out that each one of our brain cells is within 20 micrometers of a vascular supply. And in total, we have about 650 kilometers of vasculature, making it an ideal way for delivery of research tools and therapeutics, because it won't require invasive brain surgery. However, the adult brain is protected by the blood-brain barrier, a structure composed of endothelial cells that form tight junctions, reinforced by astrocytic end feet, and other cell types. The barrier allows nutrients and also blocks pathogens, such as viruses and bacteria. Specialized receptors can enable selective permeability of blood circulating agents, and we can recruit such gatekeepers to deliver therapies across the blood-brain barrier. One example that works in children with a more permeant BBB is the systemic delivery of the adeno-associated viral vector 9. In adults, AAV9 doesn't cross as strongly and transduces mainly astrocytes. The current status is that we have limited knowledge of BBB transporters, despite strong BBB biology work from pioneers such as Dr. Gu. More research is needed. The transferrin receptor, for example, is one of the strongest ways to cross the BBB. It was discovered 4 decades ago and has been used as a BBB shuttle for about a decade. Has an essential role in physiology and is present throughout the body, which could cause side effects. This is why the central question that I've been focusing on is, what are the mechanisms that enable access from the bloodstream to specific brain cell types across species? In recent work, I used directed evolution of viral vectors to cross the blood-brain barrier. Now I'm leveraging such engineered vectors for basic biology discovery, and once identified, these receptors can be used for rational engineering of delivery methods. I'm excited to recruit newly identified BBB transcytosis pathways to understand, prevent, or cure brain disease. And importantly, this paradigm can be applied to understand other physiological barriers, such as placenta, for basic and clinical biology. My solution to the problem of lack of mechanistic knowledge for BBB crossing was to take a protein engineering and directed evolution screening approach. In collaboration with Caltech alum Dr. Ben Deverman, we designed a system where we can generate diversity of AAV capsids, deliver it to the bloodstream of rodents, and apply positive selection pressure to look for capsids that can make it to the brain. Some successful results were in the form of the capsid PHPEB that has very strong presence in the brain, but one can also see that there is strong expression in the liver, which can be a problem for toxicity. Then one can go back to the directed evolution and apply not only positive selection pressure for brain capsids, but negative selection pressure to detarget the liver. In 1 decade of work, we've done iterative engineering of the AAV protein shell at multiple surface loops, which led to widespread and non-invasive delivery to the brain, while at the same time detargeting the liver. In addition to targeting different organs, we can also target different cell types. And directed evolution gives hundreds of AAVs with interesting desired profiles across organs or cell types. And what we can do is to use data science to both identify and test the most promising solutions. What we've learned was that after BBB crossing, engineered capsids can target distinct cell types, such as neurons or astrocytes or endothelial cells. And a decade of work led to a toolkit of systemic AAVs for diverse species, including non-human primates, and tools that can not only cross the blood-brain barrier, but target different cell types within the brain and also within the peripheral nervous system. These tools have been used by researchers in many fields, for example, to do comprehensive brain-wide in vivo genetic screens. Our use of the tools and other labs' use of the tools showed us that the blood-brain barrier varies across species and strains. One interesting result was that engineered vectors can cross the blood-brain barrier in certain strains, such as C57 here, but not cross in other strains. This is an important result because most likely we humans are more different than these inbred mouse strains by virtue of our biological variables, whether it's age, disease state, sex, or ancestry. This is why the engineering approach that I took initially is now naturally leading my laboratory towards a better understanding of the mechanisms underlying blood-brain barrier transport specificities, both across but also within brains. And I'm excited to look at regional vascular signatures beyond density, including transport molecules. We already have the tools to do this. My approach is rather simple. Having engineered BBB penetrant AAVs, we can just ask them, how do they get across? How do engineered vectors find solutions to cross the otherwise very stringent blood-brain barrier? We are using AAVs from our laboratory and others, and we have designed screens where we can actually look and see what are the receptor matches for such engineered vectors. This allows us to transform mechanistic studies of the blood-brain penetration from a difficult problem, a decades-long standing challenge, into a series of rather solvable challenges in receptor biology. And I'm excited to tell you about one such unexpected basic science discovery. It turns out that at least one of the engineered AAVs has found a rather unusual solution to crossing the BBB in the form of an ancient conserved enzyme, the carbonic anhydrase 4, part of the Rubisco pathway. Strongly expressed on the blood-brain barrier, this has been the solution of some engineered AAVs. And indeed, when we tested them in knockout mice that lacked the CAR4 receptor, we could see that the CAR4 binding AAVs do not transduce in the CAR4 knockout, but they transduce strongly in the wild type, while AAVs that do not rely on CAR4 can transduce both. This result is exciting because in addition to being an unintuitive solution to BBB crossing, the CAR4 also has less off-target expression versus other human BBB transporters. If we look at the transcripts of CAR4, transferrin, or the recently reported CD98, we can appreciate that in liver you have very low CAR4 expression, while in the brain is still strong. However, there is so much more to find out, and I'm excited to apply AAVs from my laboratory and others to discover basic biology. And what we can do is to perform high-throughput screens focusing on human membrane-associated proteins. And from the screens, we can take the leads and do biochemical studies such as SPR binding, and also look for transcytosis for infection of different cell types in different organs. What was difficult to address until now suddenly becomes tractable because of the diverse solutions that can cross the blood-brain barrier. I'm hoping that I can use this platform not only for the brain and blood-brain barrier, but it can be applied to other organs that have strong entry solutions through AAV engineering. We can now use structural biology that's enhanced by AI to look at the interactions between BBB receptors and their cargo, whether endogenous or repurposed. And this allows us to move into the next exciting stage of bypassing limitations of mechanism-blind screens for receptor-targeted engineering. We can do this with diverse modalities that can allow for refined tropism and also predictability across species. I am very excited to show you preliminary results that we can indeed move beyond viral vector delivery. We can dock antibodies onto the CAR4 receptor, and you can see that the CAR4 binding antibodies do cross the blood-brain barrier strongly and they enter neurons. In this case, we used an antibody that's used in the clinic for lung cancer that does have brain metastasis. We can also target different cell types, but it is critical to have knowledge of multiple receptors with different properties. Encouragingly, the two receptors we already discovered, CAR4 and LRP6, do have different biodistribution and binding properties. In addition to crossing the blood-brain barrier for therapeutics, it's important to identify blood-brain barrier receptors so we can anticipate and block pathogens that can access the brain and cause severe disease, such as HIV. Also, a bacteria evolved to use the transferrin receptor. What else might evolve? If we look at the SARS-CoV-2 genomes or other pathogens, might they evolve to have a match with blood-brain barrier receptors? I hope not. But if they do, and we know the BBB weak points, we can prepare better. What I've been doing with the group in exciting recent work was to use data science to speed up engineering of blood-brain barrier targets, not only engagement for delivery, but disengagement for blocking pathogens. And we have design methods where we can compete against peptides with candidate blockers. And at the same time, we have modalities to generate mini proteins informed by the AAV-directed evolution. And these mini proteins are highly versatile, and we have, in addition to designing them in silico, we were able to make them and experimentally validate them. I hope I convinced you why understanding BBB transport biology in diverse evolutionary contexts is important, because it will allow us to deliver brain therapeutics of diverse modalities. And once we cross the blood-brain barrier, we also have the possibility to be cell type and region specific. Understanding blood-brain barrier and the receptors will help us navigate health threats from bacteria and viruses or brain metastasis. And it will also help us cope with the reality of biological variation, whether it is by disease state, sex, age, or ancestry. My hope and dream is that we will have not one or two ways to cross the BBB, which is pretty much where we are today, but hundreds, as needed by biological variability and needs, and that this knowledge can also have us ready for brain-invading pathogens. By transforming the vasculature into a highway, we can both understand and fight brain disease with targeted access of modulators to needed regions and cell types throughout the brain. At the same time, we could anticipate emerging threats from evolving pathogens that might cross the blood-brain barrier, and be ready with blockers. And I hope we can do this in a way that is both understanding and respectful of biological variables imposed by mammalian evolution across individuals and lifespans. And it's important to know that this paradigm goes beyond blood-brain barrier. It can be applied to other physiological defense structures. Thank you for your attention, and this team, with your consideration and support, is ready to deliver.